Hi, I'm Todd Dunn, Appoquinimink School District Science Specialist. I'm here today to talk to you a little bit about our district STEM fair. Our district STEM fair was created to allow students the opportunity to engage in their own personal research opportunity or experience. This year we had representation from 11 different buildings, which included participation from over 300 students, which submitted over 200 projects. At the end of our annual STEM fair, we have an award ceremony where we give out prizes and medals to all participants. And now I'd like to take a minute so you can see some of these projects and hear from some of the judges as to what they saw. All right, so Zach, tell me a little bit about your project.
Okay, well, basically I wanted to combine my two favorite interests, music and biology, and I came up with the idea of basically swabbing my saxophone, but it's a bit more complicated than that.
Swabbing your saxophone? Like different parts of it?
Yes. A common problem in the band, and it's hard to say this, is people share instruments commonly. And that spreads diseases like wildfire. When one person gets sick, everyone gets sick, and this could be part of the problem. So basically, I wanted to look into what kind of bacteria or fungi or mold grow inside of the saxophone. And I started off with 20 blood auger plates. As you can see, this is the control right here. And the control was the saxophone, a saxophone that hasn't been played for 6 months, and the surface is pre-sterilized with alcohol swabs, so it's completely clean, and that's what the control should be. Next, I moved on to 4 surfaces. There's the reed, the mouthpiece, the brass, and the keys. So all of those are either in direct or indirect contact with the mouth, besides the keys, which are with the hands.
All right, so you had a control, then you checked the different parts of your saxophone, and what'd you find out from these pictures here? It looks like some pretty gross stuff.
The 2 parts of my experiment were 1, time, and 2, whether wiping it with a sterile cotton cloth would have any effect, because that's the common cleaning method of any.
That's the recommended.
Yes.
From the band and from the band people, they say wipe it with a cloth.
Yes, swab it out after you play it. So what we found were the first step was actually just looking at the plates and taking observations. I chose blood auger plates because when pathogenic bacteria come in contact with blood cells, they lyse the cells, either alpha hemolysis or beta hemolysis. And so the blood auger plates allowed me to see them. All of these had beta or alpha hemolysis. You can see in the dark spots right here, especially, pretty much the entire plate had beta hemolysis. And then alpha hemolysis is, for example, if it's a clear patch, that's where it completely destroys the cell. And there were a couple spots of alpha hemolysis.
So that strain of bacteria is
pathogenic.
Both are pathogenic?
Yes, both are pathogenic.
Is one worse than the other?
Alpha hemolysis is typically worse, but you don't really want either. There are common mouth bacteria that do lyse blood cells, but you don't really worry about them too much, and they can be pathogenic, but to other people. Now, as you can see, this is wiped, or this is unwiped and wiped at time 0. So this was right after playing the instrument. As you can see, the wiped had significantly less bacteria. There's still a lawn on reed and wipe, but you can actually see individual colonies, whereas this was just complete overgrowth. Also, the wiped brass had pretty much no growth, and the wiped keys had significant decreased growth compared to here.
All right.
In concerns to time, I waited 24 hours. Each were incubated for 48 hours at room temperature. But in concerns to time did decrease the bacteria significantly. But as you can see, the wiped was significantly less bacteria on the reed, about the same on the mouthpiece. There was no growth on the brass and very, very little growth here, whereas here you can see a couple of of alpha hemolysis spots and pathogenic bacteria on the keys and the brass.
So what was your conclusion? Like, what did you find out overall when you did this? Like, what did you learn from it?
there are pathogenic bacteria in the saxifrage. The next step was actually identifying them. So two of the main pathogenic bacteria that I found were Staphylococcus aureus and Neisseria meningitidis, and those can be very dangerous if they are allowed to grow and if you don't clean your horn. I identified them by gram staining.
So the point of this then is you really need to clean your band instrument properly.
Absolutely.
Even if it's your own, let alone not sharing with other people. So you decided to look at what? How caffeine affects your heart rate. And why did you do this?
Because since my mom drinks a lot of caffeine, and since she's 10 months pregnant, I was
Your mom's 10 months pregnant and drinks a lot of caffeine? Okay.
And since I was kind of worried about her, I decided to do this project to see like if you drink decaffeinated soda and if you drink caffeinated soda, if it would increase your heart rate.
Okay. So you wanted to see, did caffeine increase or decrease your heart rate? So what did you do?
I checked their pulse 4 times, so one before they drink.
Who's they? Just so you know here, who's they? Dad and mom?
Mom, my friend's mom.
Okay.
Friend's grandmother, and then my friend's.
So you had 5 people and you checked their heart rate before they drank a soda?
Yes. And then afterwards, when they drank the soda, I gave them 5 minutes to sit down and listen to relaxing music, and then I checked their heart rate again. And 5 minutes after, I had to check it again.
Okay.
5 minutes after, check it again.
Okay.
Yep.
So 5 minutes after, 10 minutes after you started, and 50 minutes, and then that's what your results were.
Yeah.
So what did you, after you got all those results, what did you find out? Like, what did that tell you?
I kind of figured out that my hypothesis was incorrect.
Okay.
It was incorrect because.
And that's fine, right? Because
yeah.
Scientists' hypotheses are incorrect all the time.
Yeah.
So it was incorrect. So what did you find out?
I found out that most of the people who had decaffeinated, their heart rate went up and then went down. But for the people who had caffeine, it was up and then it went down. So theirs was lower, then it went up, and then back down.
Okay. So if you were to do this experiment again, what would you do differently?
I would probably test them twice with the decaffeinated soda and the caffeinated soda.
So you would have each person take a sample with twice with caffeine and twice with no caffeine. Okay. And what would that do for your results?
I would try to figure out which one went up more. Was it the decaffeinated or caffeinated? So I could see which one.
Well, I think you did a good job. Very nice job.
Thank you.
Tell me a little bit about your project.
So, we intertwined these 2 books, which means folding the pages over pages, like.
So, what gave you this idea to take 2 books and take every page and alternate it?
Well, my family is a nerdy family, so on Friday nights, instead of watching movies with pizza, we watched hours and hours of scientific shows. So one night we were watching like a Mythbusters or None of the Above or something like that, and they were doing that. And
okay, so you basically saw Mythbusters or one of those cool scientific shows and said, wow, that gave me the idea to do something for a science project.
Yes.
Okay, and then your idea was to take these 2 books, put them together, and you actually drilled handles into this?
Yes. We didn't, my dad.
Okay, so your dad helped you with the safety part of drilling the handles.
Yes.
And then from here you did what?
Well,
It looks like you took this and you held it and then you added...
Yeah, so we wanted to see how many pounds it would take to pull them apart. So we put 2 10-pound barbells in, and we into the bucket, and we attached it to this bungee cord thingy.
Because our hypothesis was that we thought it would take 20 pounds to pull these intertwined.
You thought it would take 20 pounds to pull that apart?
Yes.
Okay.
Because it wasn't. It's not.
Because it was just 2 little books.
Yeah.
So how much is in here?
That's 20 pounds right now.
Okay.
But they wouldn't pull them apart. So we decided, well, maybe 30 pounds would do it. So we added.
So you added 10 more pounds.
Yeah.
So now you got 30 pounds.
And they still wouldn't come apart, and that's because of the friction.
So wait, what's holding that together?
The friction.
So the friction between those pieces of paper are more than what?
Then the 30 pounds. Yes.
All right, so let me ask you a question. How much can I put on here before it does fall apart and overcomes friction?
We never figured that out because my mom ran out of bars.
So what was the most you put in there?
30 pounds.
Okay. Okay. And then did you test this one too? I mean, this is like...
Because I already know that it would take 2 times.
So let me ask you a question. Which do you think would hold more? This one or this one?
In the state that they're in.
And why do you think this one would hold more?
Because there's like 2,000 pages.
So there's more pages, so there'd be more friction, like you just said. Okay, so that's your prediction that this one would hold more than this one. So it sounds to me like you have just created yourself a good project for next year, because you could expand on this and you could see which one takes that. We could get you some more weights, and I would be very interested to see how many weights you could put on this before it. Because like I mean, you're being honest. You didn't glue any of these, right?
No.
So all you did was just alternate pages. Okay. Well, very nice job. I'm going to be very interested to see how many weights you have to put in this bucket until that thing goes. What do you think? How much do you think it would be?
I'd say like.
Having done this experiment, what would you guess it would be around? Like, you think it's going to be another 5 pounds? You think it's going to be another 10 pounds? You think it's going to hold 100 pounds?
I think it would take like 50 or 60 pounds.
Okay. Well, I think you guys did a really good job.
Thank you.
Hi, I'm Sue Gleason. I work at the University of Delaware working with science student teachers, but I've been a science teacher in this district for almost 3 decades. Today I'm acting as a science fair judge for the annual STEM Science Fair. This has been a lot of fun. I've been going around talking to a variety of students. Primarily I've been judging middle school and high school, and watching and listening to the kids explain their research that they have done on their own. There's so many different topics. Their enthusiasm is absolutely overwhelming. They have chosen topics that are personal to them. So we have one student who is looking at behavior of his dog. What colors do dogs see best? I just spoke with a student who did some research about her hermit crabs, trying to see what stimulus affects the hermit crab. Listening to a student now who's discussing safety in football helmets. Also listen to one talk about what sunblock is good to use. The kids are excited. They've picked topics that totally appeal to them, that have meaning to them in their life outside. Questions that we ask the students, we start by asking them to explain their project. Gives them a chance to tell us what they did, why they did it, and what they've learned. They show us their data, they have graphs and charts and written documents to go with it. We often follow up with questions where we want a little more detail, and we seem to be ending with a question on what would you do next time? If you were going to do this same project, what would you change? How would you improve it? And they all have good answers for us. They see the faults and places for improvement, which is really one of the things that's great about the STEM education. It's engineering at its best.
Thank you very much for watching those clips. I hope you enjoyed hearing from the students and the judges with regards to this year's science fair. I also am looking very forward to seeing you submit your own projects next year. For more information, please contact your science teachers at your building, or if you have any other questions, feel free to contact me. Thank you.